Seeing is believing. It's a simple phrase that speaks to how much we rely on vision to experience the world. But vision itself is far from simple. It's a complex process that some estimate accounts for nearly half of the activity in the brain. Ironically, it's a process that scientists can't see well in the living eye, which is an obstacle to developing effective therapies for vision loss. But that's about to change. The National Eye Institute's Audacious Goals initiative is taking the next step to restoring vision through regeneration of the optic nerve and retina, the light-sensitive tissue in the back of the eye. Researchers funded by NEI are currently developing next-generation imaging tools to see not only the structure but also the function of the visual system, from the retina to the optic nerve to the brain. Thanks in part to Spanish scientist Santiago Ramón y Cajal, we've known the anatomy of the retina and optic nerve for more than 100 years. Using tissue samples, Cajal examined the human eye at the cellular level. He created remarkably accurate illustrations of the neurons he saw, including photoreceptors and retinal ganglion cells. Rod and cone photoreceptors in the retina translate light into visual information, while the ganglion cells that make up the optic nerve send that information from the retina to the brain. We now know that these same neurons are destroyed by blinding diseases such as age-related macular degeneration and glaucoma. To regenerate these neurons and restore vision, we need better methods to see these cells and measure their function in the eyes of living patients.
For the past decade or so, we've made great advances at seeing microscopic detail of the structure of the retina, to the point that we can sometimes see individual cells.
There is much more to see inside the retina and optic nerve, including details about individual neurons, their health, and their function. Several ongoing projects federally funded through the NEI Audacious Goals initiative have recently begun to develop new imaging technologies to do just that. One challenge to viewing the retina is that light becomes distorted as it passes through the front of the eye. That distortion increases at higher magnifications. The solution is an approach called adaptive optics, which measures these distortions and corrects them.
In a nutshell, our technology allows us to see things that the average clinician can't. So whether that's cells in a normal, healthy retina, or in a diseased retina when some of those cells begin to degenerate or change. Those things often happen in a way that's not detectable clinically, and by the time it is detectable clinically, significant damage has already occurred.
Several researchers supported by the NEI Audacious Goals initiative are pushing the boundaries of retinal imaging to get a better view. Beyond anatomical detail of the retina, researchers also want to be able to see when cells are functioning properly. That includes biochemical functions inside photoreceptors.
Those are like engine in the car. This is the most critical part of the retina that is responsible for capturing the light and responding to it, changing, translating the physical stimuli into biochemical language.
That biochemical response inside photoreceptors leads to an electrical response. Ultimately, electrical signals will pass from photoreceptors to other cells in the retina, including the retinal ganglion cells.
What we're interested in seeing is how all the neurons in the retina work together, from the photoreceptors up to the ganglion cells. Photoreceptors themselves are relatively easy to see. These other neurons are more difficult because they're transparent. What we're hoping to do with this project, using funding from the Audacious Goals initiative, is to be able to resolve these cells and measure their functional properties.
Beyond examining the retina, having a detailed view of the optic nerve is critical to understanding its health and function.
With the technology we developed in our lab, what we can do is we can image the nerve, the optic nerve, and without touching it, we can see how well the nerve function is. And also we can look at the nerve to see how damaged it is.
For the next few years, these research teams will continue developing state-of-the-art methods to observe cell structure and function in the human retina and optic nerve. Already, they've been able to observe details never before seen inside the living eye.
We're really on the cusp of a revolution in our ability to treat blinding eye disease. I think that molecular biology is generating a whole number of approaches that weren't available before, including the use of stem cells to restore vision and gene therapeutic approaches. So it's a very exciting time now, and what we need in order to move forward is to accelerate the rate at which we can deploy these technologies and make them work in curing blinding eye disease.
The National Eye Institute is part of the National Institutes of Health. For more information about the NEI Audacious Goals Initiative, visit nei.nih.gov/audacious.